So, we will continue the discussion on the differentiation assay. So, the assay that I finished in the last module is that alkaline phosphatase assay. Now, we will start with this late differentiation marker, that is the osteocalcin. So, this osteocalcin assay is typically expressed in the later stage of differentiation, and this is just a brief background on this osteocalcin. It is essentially non-collagenous protein, which is a molecular weight roughly around 5.8 kilodalton, and it contains 49 amino acids, is synthesized in bone by the osteoblast. So, essentially whenever your osteocalcin expression is upregulated, or whenever your osteocalcin expression is increased, that means that is also the reflection of the osteoblast activity, since osteocalcin is synthesized in bone by the osteoblast. Then after production it is essentially incorporated in the bone matrix, and rest is found in blood circulation. Higher osteocalcin expression reflects the rate of bone formation. So, here this is one particular assay is performed, that is immunoassay, that is enzyme amplified sensitivity immunoassay, that is called this particular immunoassay is performed in microplates and designed to measure specific protein present, that is osteocalcin biological samples. So, like before, what you do is that you first grow the cells on a substrate or a culture plate, and then osteocalcin from samples reacts with that capture monoclonal antibody, that is MAB1 as shown here. So, essentially it gets tagged here, and then comes this MAB2, that is this anti-osteocalcin monoclonal antibody 2, and their conjugate is added. This antibody conjugate then binds to the immobilized osteocalcin. So, first is that you have to immobilize osteocalcin, and then you have to add this MAB2, and then unbound conjugate is washed out during this assay, and then a solution, a specific solution is added, and the color development is proportional to that amount of osteocalcin present in the samples and the controls. And then it is essentially determined spectrophotometrically at 450 nanometer. So, these whatever assays I have discussed so far, these are typical colorimetric assays, and each assay certain optical density changes is measured at a specific wavelength using a spectrophotometer, and and then change in the optical density or color development is attributed to either specific protein or protein in the samples, or it is it is a signature of some viable cells or dead cells in the solution. So, this is except some specific details that more or less this methodology of this assay is similar to some of these assays I have discussed before. You have to first prepare the reagents, then add some specific amount of the sample like 25 microliter and so on. Then you add this anti-OST HRP conjugate, then cover the plate and incubate for 2 hours in the room temperature. Most of these procedures are essentially very standard procedures. So, there is no scope of any modification in these well established protocols or procedures that I am discussing in the context of all these assays. And finally, this osteocalcin expression is read at is determined at 450 nanometer wavelength. The fourth one, this so far I have done ALP, then I have done osteocalcin, and this is the third one, that is the Runt related transcription factor 2, or in brief it is mentioned as RUNX2. So, this is a protein in humans, it is encoded by the RUNX2 gene in humans, and this RUNX2 expression is largely restricted to osteoblasts. So, essentially as you see that all these expression is mostly related to specific cells like bone cells like osteoblast and mesenchymal condensation bones, cartilage and teeth. And this protein is essential for osteoblastic differentiation or skeletal morphogenesis or chondrocyte maturation. So, if in the context of this human mesenchymal stem cells, if you see that when they will differentiate into 3 different lineages like osteogenic lineage, like chondrogenic lineage or myogenic lineages. So, along this lineage when it gets differentiated, so there this RUNX2 expression can be monitored to confirm the how this differentiation process is progressing, as well as this RUNX2 protein is essential for the bone formation remodeling, and RUNX2 mutations are associated with this bone development also. So, what is the principle of this RUNX2? This is again based on a different assay. This is called enzyme linked immunosorbent assay like ELISA. So, this is that abbreviated form of ELISA, which biologists they use very regularly. It used to detect an antigen in a given sample, and this antigen in liquid phase is added to the culture well plate, and it adheres to the walls coated with this antigen. And so, it is a primary antibody that specifically binds with this antigen, and this enzyme linked secondary antibody is added, then reacts with a chromogen, producing a color change to quantitatively and qualitatively detect the antigen. Essentially again it is a colorimetric assay, and so it ends up with this color change, and this color change you can determine quantitatively. So, these 3 assays that I have done for this or I have discussed this module as well as previous module. So, let me summarize that. So, ALP as I said is early differentiation marker, RUNX2 also is early differentiation marker. So, mostly if you plot this expression as a function of culture time point in culture, let us say this is the ALP and this is the osteocalcin. So, suppose this is day 7, this is day 14, this is day 21, this is day 28, one would expect that ALP expression increases in the initial stage, and thereafter as you go to 3rd day or 4th day, this ALP expression may go down, and when it is going down, then one would expect that osteocalcin expression is go up. So, this circle essentially means osteocalcin, this triangle essentially means ALP. So, in nutshell what I am trying to explain to you that if one measures this ALP and osteocalcin expression at different time points in culture, one would essentially see that if there is osteoblastic differentiation is taking place, then this ALP expression would increase during the initial stage like up to 2 weeks in culture. And you keep on measuring this ALP expression because of your lack of knowledge in cell biology, then you would see that this ALP expression will decrease in the 3rd and 4th week. And during this late stage, it is the osteocalcin expression which will pick up, and this osteoblastic and osteocalcin expression will increase in the late stage of differentiation. So, that will clearly give you a signature that yes, indeed that osteoblastic differentiation is taking place, because the 2 signatures of this early ALP expression increase, and as well as a late osteocalcin expression increase. So, that is why I have mentioned it very clearly that it is a late stage marker, that is high expression on 21 to 21 days. The buffer, pH concentration, washing steps, etc., incubation period, all has to be optimized for each assay. Okay. Now, next set of assays that I will briefly mention, may not be in that much details that I have done for this earlier 5-6 assays, and this is called immunofluorescence assay. I will just cover the basic principle of these things first. So, it is an antigen antibody reaction essentially wherein these antibodies are chemically conjugated to fluorescent dyes. So, these fluorescent dyes can be fluorescein isothiocyanate like FITC. So, FITC conjugated FITC will be tagged to certain antibodies, and these labeled antibodies bind to the antigen of interest, and which allows antigen detection using either ultraviolet or fluorescent microscope. So, you can use the same fluorescent microscope in this immunofluorescence while using the immunofluorescence assays. Little bit more details, like here fluorochromes, as I mentioned before, that fluorochromes are essentially dyes, specific dyes which absorb ultraviolet rays and emit visible light, and this process is called fluorescence. There is a clear wavelength difference between these two, and this technique is based on this some of the scientists' pioneering work, which include Kunz and Kaplan and later by Mary Osborne. So, this is that your cell membrane, and then you have a primary antibody is shown here, and this is the fluorescent secondary antibody. Now, when you add these things, that cells with specific antibody will now be tagged with this primary antibody. So, this opportunity will be absent if the cell does not have that particular antigen. What is the typical excitation and emission wavelength? As you see in each case, in case of this using the fluorochromes, which I mentioned before, that emission wavelength is always greater than excitation wavelength. So, that fluorochromes will be excited at a shorter wavelength than that which it will eventually emit. So, these two things that is again you can see very clearly here. So, these are the list of different fluorochromes, and these fluorochromes, they are excited at different excitation wavelength, and it will emit different wavelength, larger wavelength, and it gives a different color spectrum. Like for example, this particular fluorochrome, which is Host 33258, then it will give blue in color. Texas red or rhodamine on the other hand will give red in color. FITC, that fluorescent isothiocyanate, that will give green in color. So, this is the different fluorochromes with different emission wavelengths will produce different colors. So, it is not possible for a student to remember the excitation and emission wavelength for so many number of for different kind of fluorochromes, but certainly those with circles one can try to remember. This is little bit more details of this protocol, which I would like to skip. Then we will go to immunofluorescence staining. So, as I have shown you before, that you have a primary antibody which is added, and in case of direct immunofluorescence, then you add this fluorochrome which will be tagged here. And in case of indirect immunofluorescence, you add a secondary antibody, and then again the same tagging phenomena will take place using the secondary antibody also. So, in the summary, this immunofluorescence is used for the evaluation of cells in suspension, cultured or microarrays for the detection of specific proteins, and immunofluorescence technique can be used for both on fresh and fixed samples. And in immunofluorescence techniques, that antibodies are chemically conjugated to fluorescent dyes, and these fluorescent dyes are shown a few fluorescent dyes are shown, and in the form of a table where that excitation and emission wavelength and then color that they are expected to develop, those things are mentioned. And other things it is mentioned here in this summary slide, that after these antibodies are appropriately labeled, they bind to the antigen of interest, which allows for antigen detection through fluorescence techniques. Okay. So, one of the other techniques which is kind of more widely used in this biomaterials, or which is now currently being used in biomaterials research for more quantification of the different cell fate processes. Now, these cell fate processes I have mentioned before, like cell division, cell migration, cell proliferation and all those things. So, for this quantification, this FACS analysis is used quite a lot. Just to give you an idea, like when you do some 2-dimensional cell culture on a sample. So, you have these cells will spread here, and these cells will now there is a 10 to the power 5 number of cells which will come, and then they will tend to adhere on the material substrate. So, this is a large number of cells, 10 to the 5 number of cells per milliliter, and then when you add this so large number of cells to a biomaterial substrate, then you will use certain assays like MTT or LDH or WST1, all these kind of assays. They will produce the results in the bulk, in a sense that they will tell you that this much fraction of the cells are live or this much fraction of the cells are dead, but you do not have any access of individual cell specific information. For example, it will not give you that exact number of the cells, that exactly this many number of cells in reality are live or this much number of cells in reality are non-living. Now, those kind of precise quantification is possible using the fluorescence activated cell sorter, where a cell suspension is made to flow through a tube, and these in this fluorescence cell sorter tube, and then a laser will focus on this particular cell suspension as it goes through the tube. Then there are two detectors, one is that FSC, forward scatter, forward scatter, and then side scatter, and both these detectors are kept at 90-degree orientation, and then forward scatter will tell you the size of the healthy or dead cells, whereas side scatter will give you the fluorescence or granularity. Now, once it goes through this column, then it will be and you use certain fluorescent dyes to start with, and then because of the fluorescent dyes you use, then it will be these cells will be attracted either towards this positive electrode or towards the negative electrode. As a result, few cells will be collected in that positive side, and other cells will be collected at the negative electrode side. So, then this particular technique is used to quantify that what is the fraction of cells are at different stages in the cell cycle, or what is the fraction of the cells are live and what is the fraction of the cells in apoptosis or necrosis stage. Now, for that first we will discuss that use of this fluorescent flow cytometer to detect the cell apoptosis and necrosis. So, typically that facts will give you a plot like this. So, as it is shown here, along the x axis is your forward scatter, along y axis is your side scatter. There will be lot of concentrated area where these dots will be there in multitude, and there is some area where these dots are very discontinuous or where this is not a very dense dots. First of all, each dot in any FACS plot contains the information from one single cell. So, essentially if you now quantify or if you now do a proper gating like the way it is shown here, so you exclude the cell debris, then you get this data. Then after this gating, if you do proper analysis to see what is the fraction of the cells lie in different quadrants. So, entire FACS plot is now divided into 4 quadrants depending on whether it is low PI expression or low annexin expression or high annexin expression. Now, to start with, in that live cells, phosphatidyl serine is typically present towards cytoplasmic side, that is towards the cytoplasmic side. Now, when cell is in necrosis stage or late apoptotic stage, then there is a flip flop mechanism which helps the phosphatidyl serine molecule to translocate to the outside of the cell membrane. Now, if you add a specific dye, then what will have, which can bind to this phosphatidyl serine, which is shown here annexin FITC. The annexin FITC can conjugate with this phosphatidyl serine. So, therefore, higher the tagging, more will be the annexin expression. Okay. So, therefore, if that more if certain region shows large number of dots with high annexin expression, that essentially tells you that these cells, this fraction of the cells have phosphatidyl serine molecule which are located now outside the plasma membrane towards the extracellular space. As a result, those cells are not live cells anymore, they will perhaps go to this more early apoptosis to the late apoptosis stage. So, this is the basis for detecting this what is the total fraction of the live cells or what is the total fraction of these early apoptosis or necrosis cells. So, propidium iodide on this similarly is impermeant to live cells and apoptotic cells, but stains dead cells with red fluorescence. So, therefore, higher propidium iodide expression will also tell you that this number of cells are dead cells. So, therefore, all the dots which shows that large propidium iodide expression essentially are not alive, and then they are essentially necrotic cells. So, each time you use this kind of fluorescent FACS analysis, these plots are to be meaningfully analyzed, so that you can extract that quantified results to essentially tell that what is the fraction of the cells are live or what is the fraction of the cells are in that apoptosis stage. Now, this protocol is fairly simple, like you incubate the cells on the biomaterial sample, and then you harvest the cells after incubation, decentrifuge and dissolve the cell pellet in serum free medium, then add different dyes. Now, these dyes would be specific for your study. For example, if you want to quantify the apoptosis, necrosis or cell viability, then you have to use that Alexa fluor 488, that is that annexin green color, or propidium iodide for these dead cells. Then after you add this dye, and again you have to incubate for the recommended conditions, and then keep the suspended cell suspension in crushed ice, and then directly add to the fluorescent flow cytometer. Okay. Now, the other things that we can see that how useful this flow cytometer is in the context of cell cycle analysis. Now, when you do typical MTT assay and all, you can say that whether this number of cells are increasing at different time points in culture. So, it will certainly indicate that when this number of cells, number of viable cells, it increases in the time of this is the cells per unit area or whatever, that whether this number of cells increases as you grow the cells for longer and longer culture duration, that certainly shows that cells are growing, that means cells are dividing. But you do not have any clue from the simple MTT assay that what is the fraction of the cells are there in different stages of their cell growth. You just have a rough idea or overall idea that simply this number of cells are growing on while being attached to the biomaterial substrate. So, if you know you know that from my earlier modules that cell cycle goes through certain checkpoints, if you remember correctly, and then it goes from G1 to S to G2 and then finally M. So, each time at the checkpoint that certain conditions are to be satisfied, then only the cell will go in an irreversible manner from G1 to S and subsequently S to G2. And if a cell is stuck in certain checkpoint, they are not able to go from to the next stage, then I have mentioned to you that the cell would essentially initiate its own suicidal mechanism and would undergo apoptosis, what is nothing but programmed cell death. Now, having said all those fundamentals, now we would like to know in a given cell population what is the fraction of the cells in the G1 stage, what is the fraction of the cells in S phase, and what is the fraction of the cells at the G2 and M phase. And this quantification is possible if you use that FACS analysis. And again, as I said before, that for each of the cell fate analysis you have to use different dyes. Now, what all different dyes are to be used for that, you can always use certain textbooks, and then which may not be very easy to remember for different cell fate processes, but the protocol remains almost similar, like the way I have mentioned here, that after standard or conventional culture you have to add some dye, then you have to incubate, and then you have to store it and then immediately put it. Now, whether you want to use these typical cell viability or necrosis and apoptosis, you have to use different dyes. If you want to study the cell growth processes or cell division or cell cycle process, then you have to use different dyes. So, essentially for each time you have to use different dyes, and these dyes once you use that dyes, then there are certain scientific rationale which will tell you that the progressive reduction of the specific dyes will give signature of certain processes. Now, in the context of the cell proliferation, you see that first generation 1 to 2, then third generation 2 to 4, then fourth generation 4 to 16. So, like that 1 to 2. So, each time if you use certain dyes which will get their intensity gets halved, like because that when a cell goes from 1 to 2, that means now this dye is absorbed by 2 cells instead of 1 cell. So, intensity if you measure, it should be gets halved. Now, whatever intensity you have measured here 2, then when it goes to 4, then it will be subsequently halved again. So, it will be like 1 by 2, 1 by 2, 1 by 2, like that the intensity will be reduced, and essentially the sequentially halving of dye will tell you that well cells are proliferating at different time points in the culture. So, this is certain rationale. You have to know little bit of biology, and you have to see that how to read this kind of data to actually interpret what kind of cell fate processes the cell is undergoing. Now, coming back to the cell cycle analysis, now you get this kind of plots. Now, these plots are essentially coming from different gating like P6 gating, P7, P9 gating and P10 gating. Now, if you do this particular analysis, this first peak will always give you that fraction of the cells which are in G1, then if you see the second one, that will give to S, and then third one is G2 and M. So, here for example, it is quantified as 75% is G1 phase, 15% in the S phase, and 9% is the G2 or M phase. Almost it is, if you add it up, it almost coming out to be 99%, close to 100%. Now, what is the other thing that I mentioned? I want to mention that during different time points in culture, if you see that if you plot one side is number of cells in percentage G1 and another side in the percentage G2 or M. Now, if you see that G1, the fraction of the G1 cells is increasing and fraction of the G2 M phase is decreasing, then you can see very well that cells are stuck somehow in the checkpoint, because so that this fraction of the cells which are present in the G1 phase increases and fraction of the cells in G2 M phase decreases. That means, cells are not able to cross this checkpoint, come to the G2 and M phase. So, if in one stage the cell numbers increases, in another stage cell number decreases, that clearly shows you signatures of that the cells are not able to divide properly over different time point in culture, and somehow cells are getting stuck and the intermediate stage either in S phase or in G1 phase, and they are not able to go to the G2 or M phase. Now, there are different dyes which are used for the cell cycle analysis, which are mentioned here, PI propydiamide. I have mentioned in the last slide that PI is impermeable to these live cells. Now, DAPI I have mentioned you in some of the earlier modules. The DAPI is the fluorescent which directly intercalates with the DNA of the cells, live cells. And then third one is the vibrant orange. So, all three dyes are used for different purposes to quantify the cell cycle analysis. Now, cell proliferation, the dye which is mostly used at CFSE and cell trace violet, and then as I mentioned to you that if you see this black one is the day 1, that is that CFSE intensity. Now, in the day 2 it is shifting towards the left side, as you see progressively, this is the day 2. In the day 3, that is the blue one, this is shifted again further left. So, all the time if you use the CFSE dyes, their intensity is going towards the spectrum, going towards the left, and in a way that is their sequential halving of the dye is taking place. That shows that this is and these are the indications that the cell proliferation is taking place. The sequential halving essentially leads to the generation of distinct peaks. So, you have a first generation, you have a second generation, you have a third generation, and you have a fourth generation, and you can see that peaks are going towards more and more left. Okay. So, certain guidelines for the flow cytometry analysis. First, you have to see the compatibility of the dye of flow cytometer laser settings like excitation and emission filters, and typically 10 to the power 6 cells per milliliter are required to acquire data, and one has to wash the stained cell suspension with phosphate buffer saline to remove excess dye and staining buffer, and you have to desuspend the cell palette in the PBS and mix well to avoid clumps or aggregate, and it is recommended to use always unstained cells as the negative control in that FACS analysis. And then other thing that I have mentioned in the dyes, these dyes are different for the different aspects of the cell fate processes that you would like to study. Other practical guidelines of this flow cytometry that use always a positive control wherever applicable, and the advantages or the clear advantages of the flow cytometer over any other existing techniques is that at least 10,000 events, 10,000 means each dot point, you remember in the flow cytometer plots are at least 10,000 events can be recorded at an event rate of 500 events per second. So, now you can understand that how fast the flow cytometer as a technique to detect all these cell fate processes. First of all, in all these conventional biochemical assays, you cannot detect or you cannot record cell specific manner or individual cell specific manner information which you can directly get in the flow cytometry. Secondly, you can get a huge number of events like 10,000 events which can be recorded with per second. You can essentially record 500 events, and data for all the samples must be acquired at a fixed voltage setting for the entire experiment. And during the data analysis, I have mentioned in one of the slides, you have to first get out the cell debris, or you have to get actually meaningful data points which shows very concentrated or very dense data points in the forward scattering versus side scattering plots. If you remember, this is your FSC, this is your SSC, or vice versa, you have this lot of these dotted points, and some of the data dotted are very discontinuous. So, wherever there is a concentration of large number of dot points, so those things you have to get it first. So, I think I will stop now and we will continue in the next.